right? But it's not a nerve cell releasing it, so it's not considered to be a neurotransmitter. But it's kind of, we have some examples of cells that are infected by viruses will release interferon that will go into the cell next to them and help to protect the cells from being infected by the virus. So when it enters a cell right next to it that is of the same histology type, without getting into the bloodstream, so this is a very local effect, is called an autocrine. Let's say it's released by an epithelial cell in the stomach and causes stomach muscle to contract more, or an endocrine cell in the stomach to release a hormone. It's still very nearby and isn't required to get into the bloodstream, but it's two different tissue types. So if it's not getting into the bloodstream, traveling a very short distance, but affecting a different tissue, then it's paracrine. Okay? We're not going to really deal with that now more than just understand the definition. We're going to be typically talking about hormones. Okay, you have this diagram on page 9 of the printed sheet. Kind of difficult to read the small symbol here, so I have it larger. Hormones are classified or grouped by the type of molecular structure that they have. So I want to go over these particular group classifications. Except for the major examples that I give, you don't need to memorize this entire list of each group, but I do want you to recognize the shape. So if I show you a symbol of cholesterol or arachidonic acid or a single amino acid, you should be able to classify it by that particular group. There are some questions on the online quiz that ask you, which of these are matched to its particular group type. But you'll either have time to look it up or it's something we will have gone over in class. So the three major groups are amino acid derivatives, all right, either a modified single amino acid, peptide hormones, large or short chains, and then lipid derivatives. So for the amino acid derivatives, remember that was also a class of neurotransmitters. So these are very small molecules. Single amino acids that have been slightly or to a greater degree modified. Dopamine is a neurotransmitter, tryptophan, norepinephrine, and epinephrine are all forms of tyrosine. Melatonin from the pineal gland is tryptophan. And norepinephrine, epinephrine, are modifications of tyrosine, including a thyroid hormone. And these are so closely related. Serotonin sometimes is included. These are molecules in the nervous system that are broken down by MAO. So you may remember that serotonin crisis is a problem if you're taking a drug, too many drugs that are MAO inhibitors. You can get high levels of any of these and have high blood pressure, high heart rate as a side effect. Okay? But these are modifications of single amino acids. Now, except for thyroxine, these all require a receptor on the plasma membrane surface, either because of size or of charge or lacking a protein channel. They are not going to go into the cell. Okay? most of the hormones we're talking about are required to bind to plasma membranes and utilize the second messenger system. Thyroxine is the only one that actually does use a protein channel. It's not lipid soluble, but it does use a protein channel to go into a cell. The next group also requires plasma membrane receptors. So these are either glycoproteins or small polypeptides. And they're going to have, 50, 150 amino acids strung along. So that's how you're going to be able to differentiate them as a group from just a single amino acid. All right, they're going to have multiple amino acids. And because of their size, they have to bind to a membrane receptor. In other words, all of these except for this one are going to bind to a receptor on the surface of the cell. They will not pass through the plasma membrane. So they require second messengers. Again, because of their size, primarily, amino acids are typically also charged at various pHs, but primarily because of their size. And then the last group, can I erase this so those in the back can see? Okay, the last group are the lipid molecules. And there are two types of lipids. One group are derived from a fatty acid known as arachidonic acid. Arachidonic acid is, let me move my picture up here. Has a ring and then two fatty acid chains. So anytime you see something like that, we're talking about arachidonic acid. And typically these form prostaglandins. Which get their name because they thought they all came from the prostate gland, and they don't. These can also be autocrine, but they can be hormones as well. For instance, prostaglandins are part of the chemicals released that help the uterine tissue smooth muscle in labor when it's contracting. They can be thromboxanes. We'll talk about thromboxanes as part of our platelet configuration. Prostacyclins. As a group, these are called eicosanoids. The other molecule that's involved is cholesterol, which has a 4-ring structure. As a group, these are known as steroids. And probably the most common steroids are forms of estrogen, testosterone, and then from the adrenal gland we have what are called corticosteroids. So if someone's using a cream because of inflammation or taking an injection for arthritis, prednisone for lupus, some kind of inflammatory condition, those are all corticosteroids. So obviously we're going to look at these in more detail, but as far as recognizing them as a class, Now, steroids being an arachidonic acid and cholesterol derivatives, eicosanoids and steroids, these are going to be soluble in the plasma membrane, and therefore their receptor will be in the cytoplasm or in the nucleus. And even if it's in the cytoplasm, it's going to move to the nucleus. Because steroid hormones actually cause DNA to be translated, to be replicated and translated. So that's what they keep in wartime. Okay, those of you that had me last semester, I promised you this day would come. I added an extra day to my lecture in Bio 430 a couple of years ago. And so we've covered this material already, but I promise I won't go any faster. This is in Maria, in the membrane chapter, that early in the textbook. So if you want to go back and read about that, that's where you would go back to rather than necessarily the nervous system, though it is a way in which many of our neurotransmitters work. So, a receptor, as I indicated up here, that is going to be for our amino acid derivatives and our peptide hormones, everything but bipyroxine. These molecules are too large and or have charges. They bind to a protein that is on the surface of the cell. This molecule never actually goes into the cell, right? So if we're talking about norepinephrine, or if we're talking about growth hormone, or insulin, it has its effect from outside the cell. It causes internal changes that can amplify this effect. All right, so one hormone binding to one receptor can result in 20 or more other molecules being activated to amplify its effect. But to get that signal into the cell requires the use of a second messenger. So I illustrated this last semester. Let's say that somebody left their, somebody left their bathtub running on the second floor, right? And it's overflowing, and it's leaking out the side of the door and out the windows, and I need to tell them to turn off their faucet. But I don't have a key to the house, and so I knock on the door. Somebody comes to the door, and I can't go in. I could maybe, but I'm polite. So I stand there and I say, guys, there's something wrong upstairs in your bathroom. And it's this little 2-year-old, and he's like, you know, hi. And he's not doing anything. So I say, could you please go get your mommy? And so he goes, and 2-year-olds shouldn't be answering the door anyway. But he goes and gets his mom, and I say, you know, there's something wrong with your bathroom upstairs. There's water pouring out the window. And she goes, oh my goodness. And so she goes and gets her older son who's handy with tools, and he goes and he turns off the water. Now I got the effect that I wanted, right? I got the water turned off, but I wasn't the one that went into the house and turned off the water. The mom wasn't even the one that turned off the water. The little 2-year-old and the mom are what we call second messengers. Okay? They get the cell to start whatever changes, enzymatic reactions that are needed to cause further changes downstream. That allows various levels of control, and as I indicated already, amplification, increasing the overall effect. So these are the two major mechanisms by which we cause proteins that can't go into the cell to cause changes. The most common is using G proteins. Okay? The first G protein pathway to be described was for norepinephrine. And two guys that processed this, won the Nobel Prize for chemistry and physiology and medicine in 1994. It wasn't that long ago. And by almost 40% of all diseases are thought to involve some disruption of G protein pathways. So you can see that this becomes really important clinically and in a lot of the pharmaceuticals that are developed to affect G protein pathway. Here, there are still second messengers that are involved to a degree that they're kind of sidestepped, and this is a little bit more directly. The receptor itself becomes more directly involved than in this pathway. So let's finish, sure we may not get through all of it, but let's get started on this. We're going to look at a common pathway for G proteins, and these will start on page 15. So if this common pathway, everything follows through with this. On a heads up, there's going to be a lot of new names, all right, for these, today and Thursday, for these different concepts and molecules. Phospholipase C, and phosphoinositol, and inositol triphosphate, and diacylglycerol, and phosphodiesterase, and so on, phospholipase C. So start getting a handle on it, all right? Don't write up a flashcard and a name and memorize it. First, recognize its role. What is it doing, okay? And then start to associate, okay, phospholipase C, I'm going to also have IP3 and DAG. When I have cyclic AMP, I'm also going to have the enzyme that breaks it down. So you start to put them into groups where they function, all right? That's not the end. You're a little bit involved in the complexity of mechanisms. Now the next thing is, okay, well here I use cyclic AMP. What's a role that's similar to this pathway? Which ones involve calcium? So you start doing some layering, all right? What do these pathways have in common? What's different between these pathways? So that you're not just learning each of them in isolation. So let's look at what they have in common. This is the Nobel Prize, this is 2012 here. That was a different concept of it. The first one, first presence of G protein was actually 1994 for norepinephrine. Last year's chemistry one was for the receptors that were involved. Okay. So what I did, I'm going to diagram this on the board. All right. So what we're looking at first is a lipid bilayer. What you're seeing here is the phosphate group and the fatty acid tails. And then we're looking at a transmembrane protein that goes throughout the entire membrane. This is outside the cell. So this is our receptor. Outside the cell is the site that it uses to recognize the hormone. And remember, that's why we use proteins, because of the folding. Strings of amino acids can get complex shapes and recognize other shapes. On the inside of the cell, we have our G protein. Okay? It typically has three components, alpha, beta, and gamma. And we typically look at the alpha. The alpha can be both an excitatory or an inhibitory, depending on the cell. It can increase the amount of our second messenger, or it can decrease the amount of the second messenger. Beta most commonly is an inhibitory, and we talked about it a little bit with neurotransmitters last semester, but we're not going to deal with it now. We're going to look at the alpha. All right? Here's our receptor. And bound to the receptor is the G protein. Some diagrams will show the G protein next to the receptor. This one happens to show it over here. So I'll go along with the diagram that we have, and you'll notice that when the receptor, when the G protein is bound to the receptor, and the hormone is not, that we have GDP attached to the alpha subunit. That's the inactive form. You've heard of ATP? And GTP is very similar. It's an energy storing molecule like ATP is, only it's guanine instead of adenine. All right, yes?
So the G protein is all three of those?
All three of them together. Yes, the G protein complex. So, the GDP, or the guanine diphosphate, it's already given up one phosphate, so it's in a low energy formation. And that's the first step. All right, so this is no hormone present. The hormone is absent. Then a hormone comes into play. It diffuses out of the bloodstream and is in high enough concentration to match up to its receptor. Now the hormone binds to its receptor, and that interaction, that chemical interaction of the hormone to its receptor changes the shape of the receptor. It physically isn't the same shape anymore. And because of that, the G protein no longer fits into its binding site. So it dissociates. Now on this particular diagram, and that's some of the problems with illustrations, it shows that the alpha subunit stays in the plasma membrane, okay, and just floats. Remember, this is a fluid lipid bilayer. But that alpha subunit doesn't have to stay in the plasma membrane. Okay, it can come out of the plasma membrane. It's often drawn as staying in the plasma membrane because its activity is to bind to another enzyme, that is already in the plasma membrane, but it doesn't necessarily have to stay there. And now that it's free of the other components of the G protein complex, its GTP is replaced with its GDP is replaced with a GTP. All right? And that activates it. So that's going to be reconverted to a phosphate group and activate the alpha subunit. So that's a temporary situation. So that's the common pathway. Now obviously a cell is going to have more than one receptor. So more than one hormone is going to be binding to a receptor, and we talked about upregulation and downregulation, so we can increase or decrease this overall effect. So if we go back to this previous slide, there's supposed to be a movie there, but no longer works anymore. The three different mechanisms by which that alpha subunit works are going to be to work on these steps here. All right? So the first one we're going to look at is cyclic AMP. Then we'll look at calcium. And then we'll leave you completely confused by looking at phospholipase C. When we come back on Thursday, I'll review that with you. All right. So, the first thing that we're looking at, and the first one that was discovered by a scientist, was the effect of this active form of the alpha subunit is going to be to interact with the plasma membrane bound enzyme. This one is stuck in the plasma membrane. And this is an enzyme known as adenylate, or it's not wrong. Sometimes it's called adenylyl. Okay. Adenylate cyclase. Now again, remember, ASE means it's an enzyme. Not all enzymes end in ASE, but if a name of a molecule does end in ASE, then it is an enzyme. If that makes sense. This enzyme is in its inactive form. It's not going to work as an enzyme until our alpha subunit attaches to it, and the ATP, the GTP, gives off an energy form, and its attack remains as GDP. All right, now this is an active enzyme. So once the adenylate cyclase is activated, it provides its energy to the adenylate cyclase. Once the alpha subunit is activated, it provides its energy to the adenylate cyclase, and now it's active. So if we see it looking like this, because the previous form is not going to be there for very long. So this is now an active enzyme, and its role is to take ATP and convert it to cyclic AMP. So it's going to bump off two phosphate molecules and turn it into a ring structure. So now in its active form, the result, ATP binds to adenylate cyclase, and the enzyme activity is to kick off those two phosphate groups and make cyclic AMP. Cyclic AMP is in the cytoplasm. This is our second messenger. It can move around inside the cell. Okay. The adenylate cyclase is not the second messenger. It's part of the step of activating the second messenger, and cyclic AMP is now the second messenger. So as long as the alpha subunit, bless you, is bound to the adenylate cyclase, it can keep on churning out cyclic AMP. And the more receptors we have that bind the hormone, the more adenylate cyclase is activated, and the more cyclic AMP is available. All right, so that's the amplification I was talking about. Just one hormone can make lots and lots. One hormone molecule can make lots and lots of cyclic AMP. Now, cyclic AMP by itself is just one of a series of steps. Cyclic AMP isn't running up the stairs and turning off the water. Okay, cyclic AMP is the mother telling the son to go turn off the water. And we actually can have a variety of steps. So what cyclic AMP does is it further activates enzymes known as protein kinases. And in this case, they're known as protein kinase A. So without cyclic AMP's intervention, protein kinase is not active. When cyclic AMP interacts with it, protein kinase A becomes active, and its role is to add, typically, I'll put in parentheses, remove, because sometimes that's what it does. But usually, in most cells, it will add phosphate or phosphate groups to other molecules. So there may be other enzymes in the cell, and they have to be activated by protein kinase. So this is that cascade of events that start because the receptor is binding its hormone. So here's what I wrote up on the board down at the bottom for those of you that. So here's our second messenger. So we have a whole bunch of enzymes in the cell that don't work unless they have a phosphate group on them. And that is what the protein kinase does. Okay? in this case, it's particularly looking at a particular hormone, so not every hormone is going to cause glucose to be released. But in this particular example, that was the case. So now when we stop this from happening, what are several things we can do? At the very beginning, we can get rid of our hormone. Okay? So we can either break it down, we'll talk about that a little bit on Thursday. We can stop making it, stop synthesizing it.